All right, good afternoon, everybody. This is a video about a lab that we call the Which One's Alive lab. You're watching it probably because you were absent or you're online or something like that. So, in the course of this lab, we give you two test tubes, and the test tubes are labeled A and B. I don't know if you can see that clearly. Actually, I'll switch slides there. Now you can kind of see a close-up of what's inside the two test tubes. The purpose of the lab is for you to prove which one is alive. I don't care if you can guess. I hope you have some guesses as to what's inside of them, but now you have to be able to prove what's inside of them. So the first thing that partners do is they brainstorm, well, what are characteristics of life that we could test for? Now, you could test for a brain, but I'm telling you that neither one has a brain. So what are the more basic characteristics that all living things have in common? Well, what we agreed in class was that all living things have cells and DNA. So those are two things that we can test these test tubes for. But there are some other things that all living things do. And so we're going to do one of those experiments as well. So something that other living things do, they grow, they reproduce, and they use energy. And so we're going to use that all in one lab here. So I'm going to rush you through 3 labs. It won't take very long. But let's start with the growing one because it takes some time. We're going to use this machine back here. It's called an incubator. It's basically like a warm refrigerator, and it's where these guys can grow. So I'm going to start with the procedure for getting them to grow and reproduce and use energy, which means eat. So for the procedure, we have little procedures here. So it says add most of the contents of one test tube to a baggie. Oops, I left the baggies over here. Hold on. Right there. Okay, so I've got the baggie. So it says add most of the contents. So I'm going to add test tube A to this baggie. Piece of cake. There it is. Okay, and I'm going to label it A so that we know that this is test tube A. Oops, I made a mess. Okay, so this is test tube. This is baggie A, and it's got an A on it. So it says add 20 milliliters of warm or cool water, but not hot water to it. So here's my 20 milliliters of water that I've added to it, and I'm going to squish it around a little bit. It says add a decent scoop of table sugar. So here's my table sugar, and I'm going to add a scoop of it. Just enough. There we go. Oh, I'm spilling it just enough for if it can eat to be able to eat. And then I'm going to smoosh it all around. Give me just a second because I got to grab a lab supply. Hold on just a moment. All right, sorry about that. So we've got A ready to go. And then it says mix the contents, repeat steps 1 through 4 for the other test tube. And I'm just going to squish out any air that might be in it. So it doesn't have any air in it right now. It's ready to go. All right, I'm going to go ahead and label the next one. Give me just a second here. I'm going to lower it so you can see what I'm doing. So I'm going to label the next one. I'm going to label it B. And I'm going to go ahead and pour the contents. Here we go, my cap. I'm going to pour the contents of B into it. And I'm going to add some water like it said. Here's my water. I'm going to add some water into it. Mix it up, squish it around a little bit. And then I'm going to add some table sugar to it. So a scoop of table sugar to it. So here we go. Here's my bag. Here's my table sugar. Mix it in. Smoosh it up for a minute or so. Make sure that sugar gets all around. And we're ready to go. We're going to go ahead and place both bags in this machine that's called the incubator. And we're going to place it in the incubator for about half an hour to 45 minutes. So I can work on the other labs while I'm waiting for these guys. So I'm going to open it up. I'm going to check the temperature in here. The temperature is supposed to be around 37. It's 35. That'll be perfect. So I'm going to lay A and B down. Oops, I'm going to lay them down flat. I don't know if you can even see them in there, but they're in there, and we'll take a look at them in about half an hour to 45 minutes. Let's close them up, and let's get started on the next one. Let's go ahead and experiment to try to see if either of them has DNA. So I don't know if I have all my supplies that I need, but we'll try it. So I got to grab my directions. So for DNA, it starts out exactly the same way. It says add all of a sample. Let me pause for just a second. All right, sorry about that. I'm just trying to get all my materials together. There's a lot for these labs. Okay, so I have 2 bags. I labeled them A and B. This is for my test for DNA to see if either one has DNA. So again, we're going to put all of a sample in. So here's sample A. I don't know if you can read that it says A. I'm going to put that in the bag that says A. And then I'm going to put all of sample B in the bag that says B. So sample A and B are in their 2 bags. Okay, so next it says add 10 milliliters of water and a small scoop of meat tenderizer. So this is the meat tenderizer. You buy it at the grocery store. It's for if you're making some kind of a meat and you want it to be not quite so chewy. This helps loosen it up. So here's a scoop of meat tenderizer, and I'm going to put it in each one. And then I need to add some water to it. So there's some meat tenderizer in this one. I'm going to lower it so you can see what I'm doing a little bit better. And then let's add some meat tenderizer into this one. Add a little meat tenderizer. And then I'm going to go get some water. Let me pause it while I grab some water. Okay, I'm back. I've got the water. So here's bag B. I'm going to add some water. And then it says to mush it all up. So I'm mushing up the water and the meat tenderizer and the sample in B that we're trying to prove if it's alive or not. So for this experiment, we're trying to see if it has DNA in it. So I'm going to smoosh it for a bit. I need to do the same with A. So I'm going to set B down. And I'm going to grab A and add water into A, and then I can kind of smash them both up at the same time. Again, we're trying to find DNA to see if either of them has DNA. So I'm going to squish them both at the same time. Like this. There we go. I think the meat tenderizer, what it does is if there are cells present in A or B, it breaks the cells open and allows the DNA to kind of pop out of the cells so that we have better access. And I hear that this is a pretty important stage to make sure that they're really well mixed up and that meat tenderizer has had a chance to break up the cells. That's what makes meat softer or less chewy, is by adding this meat tenderizer. It breaks up the cells, because remember that meat comes from animals and they have cells. It breaks up their muscle cells and makes it a little bit softer for chewing. Okay, I'm going to hope that that's enough. So then it says add 1 milliliter of dish soap to each bag. So I've got my dish soap right here. So here's my dish soap. And I'm going to go ahead and just give it a squirt. We don't need to measure carefully, just a squirt. Good enough. And we're going to mix it up a little bit. So we've got some dish soap in there. I'm going to zip it shut again, so no dish soap goes squirting out. I'm also going to squish up B as well. So let me add the dish soap to B, and then we can squish them simultaneously. So here's the dish soap for B. There it is. And now I'm going to squish it up. I'm going to zip it shut. And now I can squish both of them, get that soap kind of mixed in where it belongs. Get them really nice. One of them is a little bit thicker than the other, but we're getting them pretty good. That next thing that we're going to do is we're going to make a filter with gauze. I have the gauze right here. And we're going to make a filter, and we're going to filter out any solid pieces so that all we get is the liquid passing through. And I will tell you that that's a lot harder to do when you don't have a partner. So hopefully I won't make a total mess out of it, but you can laugh at me if I do make a total mess out of it. Okay, those should be mixed up enough. I think we're ready to go. So let's start with A. I'm going to go ahead. Here's my beaker. I'm going to see if I can lower my camera enough. I hope you can see this. Get my papers maybe out of the way here. Okay, and I'm going to make a filter out of gauze. So here's my beaker. You don't need my Sharpie there. Here's my filter. And I'm going to cut a little hole in my baggie. And it's going to drain. I hope I don't make a big mistake and make a huge mess. That'd be kind of embarrassing. So I'm going to cut off the end right here. And then I'm just going to drain it onto this. I hope you can see this okay. Oops. There we go. It's working. It's working. I did B instead of A. I think I said I was going to do A first, but I did B first. And it's making a nice filter for me. Filtering out all the solids. Kind of hard to do by myself, but I think we've got enough. So now I'm just going to squeeze it. Get that last little bit in there, and I'll show it to you. That's our liquid that we've got. So there might be DNA in there, and that was baggie B. So I'm going to keep that set aside, get it out of our mess. It makes such a mess here, so I'll get this out of our way. Now let's do the same thing, but for sample A. So here's a clean beaker and some gauze. Here's sample A, and I'm going to cut one end of it. It smells kind of good because it smells like the dish soap that I was using. So it's got kind of a nice fresh smell. All right, I'm going to try to pour A in there. What we're trying to do is get the liquid to pass through, but not the solid. We don't want the solid to go through. Okay, and I feel like we're doing pretty well. I think we have enough liquid at this point. I'm going to squish this a little bit. Yeah, we've got plenty of liquid there. We got tons of liquid, so we're good with that. So now it says that we need to add them back into their test tube. So here's test tube A and B. So I'm going to pour it halfway up to B. So we pour it halfway up so that this test tube is halfway full of the sample that might have DNA in it. That feels like about halfway to me. All right, so that's sample B, and then let's do for sample A. Oops, that's a B again. I need an A. Here we go. This is A. We're going to fill it halfway to the top. There we go. And now the instructions say that we need to add ethyl alcohol. My ethyl alcohol is right here. This is nothing anybody would want to drink. It's very bad for us. And we're going to add it to the two samples. So here's my B. I'm going to add the alcohol. It's very cold alcohol, and I'm going to add it. And then I'm going to let it sit for a moment. The alcohol is attracted to DNA, or the DNA is attracted to the alcohol. I don't care which way we say it. And the DNA will start lifting up out of the sample if there is DNA present. So it takes just a moment while it lifts up. And I'll see if we have any. Maybe I'll let it sit for just a moment and we'll get ready for the next section. The last experiment that we're going to do while we're waiting on this one is looking for cells. So we need 2 microscopes for that. We're going to make 2 slides and we're going to look under the microscopes. So let me bring you over so that you can see. I need to pause one more time because I got to grab 2 more samples. I'll be right back. Okay, so right now I'm over by our microscopes. We need to plug them in. They're not ready or anything. And I have 2 more samples of A and B. I'm going to put just a few grains on a microscope slide. And then I'm going to add a dye that's called methylene blue. That's this dye. And methylene blue sticks to cells and makes them kind of a bluish color. And then I'm going to add a cover slip to each one, this little plastic cover slip. It protects the microscope in case our lenses bump into our samples. So let's see where you can see me. I'm going to put it right here. And I just want to put, 3 or 4 little grains. It doesn't need to be a lot. So that's that one. And I'm going to add the blue dye. And we're going to look at what it looks like under the microscope. There's the first slide. So we've got one that's ready to go. And now we're going to do the next one. This is sample B. And again, I only want a few grains under the microscope. It looks like, oops, I added way too many. I had about 10 grains there. Okay, and I'm going to add a drop of methylene blue to it. And then put a cover slip on top. And then we'll take a look at those under the microscope. All right, I'm going to pause here so that we can actually see what they look like under the microscope. All right, I switched slides so that you could see what it looks like under the microscope. And my image right now, my video is in front of one of them. So test tube A over on the left of the screen there, it looks like rocks, basically, rocks that haven't been stained correctly. And that's under low power, so they're really big rocks. Whereas, let me move my image here. Over here, these tiny little circles, those have been stained by the dye, and those little circles are cells, people. That's what cells look like. And so we've shown that test tube B has cells in it. Now let's take a look and see. Oops, I almost knocked things over. Let's see how our DNA experiment turned out. I'm going to hold them up and see if we can get the camera to be able to see the difference. So if there's anything floating inside of it, it's DNA. If there's nothing floating inside of it, then there's no DNA. And I hope that you can tell that B is full of stuff floating in it. and that's DNA. A has nothing floating in it. It's completely empty in the top because there's no DNA. So now we have two pieces of evidence that B is alive. It has cells, it has DNA. And now it may have been too early, but I'm going to check our incubator. If it doesn't look good, we'll check back. I'll pause the video and we can look in 10 or so minutes. Let's see if there's any difference between the two samples when they've been fed. Okay, so let's take a look. Bringing it in. Oh, they totally look different. Can you see that? Look at A and B. Can you see that B is all full of bubbles? It's all puffy. And A just looks like it always does. I hope you can see that on the camera. Okay, so we've shown now again a sign of life. B ate the sugar that we gave it. It started to reproduce, it started to grow, and it started to produce waste, which are those bubbles. So we have more evidence for life. All right, everybody, that's it. Let me know if you have any. Oh, and I was just going to show you a couple quick images on this. Hold on. Let me back up. This is the DNA if you wanted to see it up close. So there's B with some DNA, and then A had no DNA in it at all. And then obviously, whoops, the two bags. I'm going the wrong way, everybody. There we go. The two bags had, one was full of bubbles and one wasn't. So this baggie here was full of bubbles. All right, that's it. Have a great day.